Good evening, students. Today tips. Studies are several types. Like we can study for knowledge. Otherwise, we can study for exam point of view. Then we can study for competitive examination. Now you are studying to crack competitive exam Neet. So first you must know the syllabus in biology. Then you should understand the concept of each chapter on the topic. Then prepare a question bank in MCQ format. Finally, revise it. So continuous revision supports to get a more mark. So you must understand, first you should know the syllabus. Then you should understand the concept. Then prepare a question paper in MCQ format. Finally, revise it because revision should support to get a more mark. Clear? Then another one important point is if you study 1 to 1.5 hours, so you must take a rest minimum 5 to 10 minutes. So after 1 to 1.5 hours study, you must take a rest. Rest means rest for your brain. Rest for your brain. So after 1.5 hours, you must take a rest. Rest means you can think about other things apart from the study. You can chat with your parents or you can watch a TV. Otherwise, you can chat with your friends only 5 to 10 minutes. So after 5 to 10 minutes, your brain will refresh it. So after that, you can continue your study. Clear? So in this method, you can follow it. Easy, the brain will easily uptake your points. Clear? Okay, let's start our today topic with linkage. Linkage. So yesterday we discussed a lot of things in Mendelian inheritance, monod, dihedral cross, or etc. etc. Today topic is starts. Today I am going to start topic with the linkage. So what's the meaning of linkage? Linkage means linking. So closely arranged. Linkage means closely arranged. So the linkage is first seen by the Batson and Punnett in sweet pea plant. Linkage is first seen by the Batson and Punnett in sweet pea plant. Lathus odorous, that is a botanical term for the sweet pea plant. That question may ask in NEET, who very first seen the linkage in which plant? So you should know that one. So Batson and Punnett, they only very first seen the linkage in sweet pea plant. So definition for linkage. So phenomena of certain genes staying together during inheritance through generation without any changes or separation. This is called linkage. So while the particular genes or chromosomes will be transferred on to next generation. So at that time, genes also transferred without any changes, which is called as a linkage. Why these things are happens means due to the location of the same chromosome. That gene will be present in the specific chromosome locus. For example, that particular gene, one gene is present in the 11th chromosome means that if the 11th chromosome is transferred from one to next generation means that gene also transferred along with the chromosome. That's why I'm saying this is due to the location of the same chromosome. Clear? This is called a linkage. The next one is a sex linkage. Sex linkage, very first discovered by Morgan in Drosophila. This is also important Neet question ma. Discovered by Morgan in Drosophila. So what's the meaning of sex linkage means? Some somatic characters, genes of, so you know very well, autosomes, allosomes, two things are there. Some genes are responsible for making the body characters. Remaining genes are responsible to make a sex characters. Clear? So the somatic character genes are present in the sex chromosome, termed as sex-linked genes. Again, I repeat, genes of somatic characters which is present on the sex chromosome, which is called as a sex linked gene. That whole phenomena is called as a sex linkage. That phenomena is called as a sex linkage. So what are the types of sex linkage? Sex linkage means sex linkage broadly classified into the two types only, because you know very well the two types of sex chromosomes only available. What are they? You know very well. One is a X, another one is a Y. So the sex types of linkage also classified into the two types. One is a X linkage, another one is a Y linkage. X linkage means some somatic character genes. Somatic character genes will be present in the X linkage. Will be present in the X linkage. I mean X chromosome. X chromosome. X chromosome, which is known as a X linkage. So genes of somatic characters. Genes of somatic characters will be present in the X chromosome, which is called as a X linkage. Similarly, the genes of somatic characters, if genes are somatic characters located in the Y chromosome, which is known as a Y linkage. Clear? Somatic character gene is present in X chromosome means X linkage. If it is present in Y chromosome means which is called as a Y linkage. Next, X linkage. First one, X linkage. S linkage means X linked characters. X linked characters may be transferred from both gender, male to male or female to female. Clear? So genes of somatic characters are found in X chromosome. That inheritance of X linked character. Listen, ma. Inheritance of X linked character may be through the male and female. Male and female, not or ma. That inheritance of X linked character. X linked character will be transferred. It may be male and female. Example, color blindness, hemophilia. Those are the two examples of a X linkage. So here character will be transferred through the both male and female. That is called as a X linkage. That examples they may ask in exam. Next one is a Y linkage. Next one is a Y linkage. The genes of somatic characters are located in Y chromosomes. That is called a Y linkage. Just now I told genes of somatic characters are located in Y chromosomes. That is called a Y linkage. That inheritance of such type of character occurs only through the male. That is important one ma. That characters occurs only through the male. Such type of character is called a holocentric. Such type I underlined in green color ma. Such a type of character is called a holocentric character. I think already that question is asked in previous NEET exams. That is also important ma. So what's the meaning of holocentric characters? That Y-linked characters will be present in male. It will be transferred to another generation, another male through the male only. So male, the character will be transmitted from male to male. That is called as a holocentric one. So simply we can say character will be transmitted. Y-linked character will be transmitted from male to male. Why what is called as a Y-linked? Why? Where it is a present. The female having only the XX chromosome. homozygotic. Clear? Male only consists of a heterozygotic. I mean X and Y. Y will be present in a male only. Y sex chromosome will be present in the male only. That's why that Y-linked characters will be transferred from male to male. So male to male transferring character, that is called as a holondric characters found in only the male. That question is important, ma. For example, hypertrichosis. Hypertrichosis, that is also important. Hypertrichosis means hairy ears. If you see the some aged people, aged man means that hairs will be present in the ear of pinna. In case the father having that ears in the hairs in the ear of pinna means it will be transmitted. The son or grandson ear also having the hair. Clear? So that is called as a hairy ears. So hypertrichosis means hairs present in ear of pinna. So that is called as a example of a Y linkage. Clear? Next one is types of inheritance of sex-linked characters. Types of inheritance of sex-linked characters. First one is criss-cross inheritance. First one is criss-cross inheritance. Sorry. Criss-cross inheritance. Criss-cross inheritance. Criss-cross inheritance. Male or female parent transfer a X-linked character to grandson or granddaughter through the progeny of opposite sex. Criss-cross. What's the meaning of cross? Cross means randomly transfer the particular one. Cross means randomly. Clear? So random means in human being only two gender is available. The third gender transgender. That is not important one. That is the exception case. The majorly two gender is available. One is a male, another one is a female. Here, so that character will be transmitted from male to female or female to female, like that. Again, I repeat, X-linked characters to the grandson to or granddaughter through the progeny to the opposite sex. Like character may be transferred to the father to daughter, then daughter to grandson. Otherwise, mother to son, or mother to son, then son to granddaughter, like that. Male, female, then male. Otherwise, female to male, then female, like that. That is called as a criss-cross inheritance. What's the example for criss-cross inheritance? Means X-linked inheritance. X-linked inheritance are example for the criss-cross inheritance. X-linked inheritance, just now we discussed. The next one is a non-criss-cross inheritance, the last one. Non-criss-cross inheritance. Opposite to criss-cross. That is called as a non-criss-cross inheritance. Here, male or female parent transfer a sex-linked character to the grandson or granddaughter through the progeny to the same sex. Important word, ma. Again, I repeat, the non-criss-cross is a opposite to the criss-cross inheritance. Here, inheritance, male or female parent transfer sex-linked character to the grandson or a granddaughter through the offspring of the same sex. I mean, the character may be transmitted from a female to female, then female, like this. It will be goes so on. Similarly, either the character may be transferred from the male to male. Clear? So like that. So character may be transferred to the same sex, grandson, father, son, grandson, like that. Otherwise, mother, daughter, granddaughter, like that. Clear? So if the character is transmitted from female to female, that is called as a hologenic. That is called as a hologenic. That is also important Neet question mark. Kindly note it. Kindly note it. So that non-crisscross inheritance are broadly classified into the two types. What are they means of? First one is a hologenic. Second one is a holocentric. Hologenic means that character is transmitted from female to female. That is called as a hologenic. Example, mother to daughter, daughter to granddaughter. Clear? The second one is a holocentric. So holocentric, just now we discussed it. Male to male. That character will be transferred from male to male. Such a phenomena is known as a holocentric one. For example, that character may be transmitted to father to son, son to grandson, and so on. Clear? So, for example, holocentric example, Y-linked inheritance. Just now we discussed it, Harry years. Clear? So that's all about the non-crisscross inheritance. Next one is a sex-limited characters. Sex-limited characters. The characters are present in one sex and absent in another sex, but the genes are present in both the sex, and their expression is depends on the sex hormones. I think we know the very well about the sex characters, sex limited characters. That characters are present in one sex and absent in another sex, but genes are present in the both sex. For example, androgens. For example, androgen. What's the meaning of androgen, ma? I think everybody knows androgen. ANTROGN. Androgen is a sex hormone. Will be present in both sex, male and female, but expression level will be vary. In male, androgen hormones will be expressed high quantity, but in female, expression level is very low quantity. Clear? Very low quantity. So characters are present in one sex and absent in another sex. Absent means androgens are absent in a female one, present in the one sex. I mean male one. Clear? But their genes are present in both sex. Means androgens producing genes. Androgens are present in the both male and female. Clear? Androgens are present in the both male and female, but absent in female, present in the male. Clear? That expression is depends upon the sex hormone. Expression means that hormones, androgen hormones. The androgen hormones, excessive level of androgen hormones results are mustache, beard, etc., etc. Mustache and beard will be grow in male only. We can see only the male, but some female, in some female grows mustache and beard also. That character is called as a secondary sexual character. That character is a secondary sexual characters. Why the secondary sexual characters are present in the female means that androgen hormone level is higher than the normal level of androgen hormone. Higher than normal level of androgen hormone secretion. Clear? So that's why that secondary sexual character will be expressed. The next one is a gene interaction. Next one is a gene interaction. So the gene interaction is called as a comes under the post-mentalism. Post-mentalism. What's the meaning of post-mentalism? So mental, he said that gene is responsible for all the DNA only capable of transferring the character from one to next one. The single gene is responsible for determining the character, etc., etc. But post-mentalism, that is also like an exception case. Post-mentalism means that is also like an exception case. For example, one gene may kill the another gene. Otherwise, one gene can control the many character. Otherwise, many alleles can control the single character. Clear? So like that. So that is called as a post-mentalism. Why the post-mentalism is occurs means due to the gene interaction. You know very well, interaction means one will be react with another one. Clear? That is called as a interaction. For example, now I am explaining. Here there is no interaction. In offline classes, we can interact. We can interact lot of things. So if I ask any questions, means you can give any answer. If right, wrong, that is not important. In case right means I will appreciate you. If wrong means I will give the reason for that one. Like that. That is called as a interactions. One will be react with each other. That is called as a interaction. Here, interaction will be occurs among the genes. That is called as a gene interaction. The effect of one gene modified by the several or modified by single gene or maybe a several other genes. That is called as a gene interaction. So after the gene interaction, what will happen means a normal phenotype character will be changed. So the modification of normal phenotypic expression of genes due to the alleles are a non-allogenic one. Alleles are a non-allogenic one. So gene interaction means modification of normal phenotypic expression of genes due to their allele or non-allogenic genes. Why the gene interaction, where the gene interaction will be takes place means due to the presence of the allele or maybe a non-allogenic gene. The gene interactions broadly classified into the two types. What are they means? Intragenic interaction. Another one is intergenic interaction. One is intragenic. What is the meaning of intra? Intra means within. That is called as intra. Inter means outside. For example, in your school, maybe conducted a intra-school competition. Means that competitions may be conducted within classes. For example, various classes. For example, competition may conduct from to the 11th and 12th of the same school. That is called as a intra, that within. Clear? In case that competition is conducted from your school or nearby your school, means that is called as a inter. What's the meaning of internet? Internet means we can get it any sources. So internet means the one will be interacted with the another one, outside one. Clear? So inter school competition means that competition occurs between the various schools. That is called as the inter. Intra means within the competitions may be conducted within your schools of different classes. That is called as the intra. So intergenic interactions. So here intergenic interactions means two alleles of genes which are present in the same gene locus on the two homologous chromosomes. Two homologous chromosomes will react to produce a modified phenotype. Just now I told here that interactions will be occurs and the alleles of the same gene. Alleles means a two alternative form of genes that the genes, the alleles of the same, the interactions will be occurs in the same alleles of the genes. So at that time, what will happen? That phenotypic. At the time, what will happen? It's modify. It's the phenotype. Modify. It's a phenotype. Clear? So in a particular gene, particular gene is responsible for to select that particular character. Means in case that interaction is occurs in the same gene, means all genes of allele. In case that interaction is occurs in the genes of allele, what will happen? It modifies its a phenotypic character of a organism, which is called as a intragenic interaction. Clear? We will discuss one by one. Intragenic interaction. So that intragenic interactions are furtherly classified into the 4 types. What are they means? Incomplete dominance. Second one is multiple alleles. Third one is codominance. Last one is lethal genes. Lethal genes. That lethal genes. So far we'll discuss one by one. First one is incomplete dominance. Incomplete dominance already we discussed. Incomplete dominance, multiple allele, codominance. Everything we discussed under the topic of exception of mentalism. Clear? Incomplete dominance. What's the meaning of incomplete dominance, ma? For example, pink color flower. So when that the flower, white color flower is crossed with the red color flower, at that time pink color will be expressed. That is called as a incomplete. The mixed color, otherwise new character will be formed. That is called as a incomplete dominance. Already we discussed. Pink color flower is an example for the incomplete dominance. Then multiple allele. Multiple allele means many, 2 or more alleles can control the single character. That is called as a multiple allele. So 1, 2 or more alleles present it, which controls the one character. That is called as a multiple allele. For example, human ABO blood grouping. That thing also we discussed. The next one is a codominance. Codominance means that both character will be expressed in the progeny. That is called as a codominant. For the same example, ABO blood grouping and coat color of a human being. Coat color and blood group of coat color of animals and the blood group of human being. That is an example for the codominance. The next one is a lethal gene. What's the meaning of lethal? Lethal means a toxic one. Lethal means a toxic one. The genes which causes death of individuals in early stage. The particular gene can cause death of individuals in early stage when it comes in homozygous condition. When it comes in homozygous condition, which is called lethal gene. Again, I repeat, genes which causes death of individuals in early stage when it comes in homozygous condition is called lethal genes. It may be dominant or may be recessive in both, but mostly recessive responsible for the lethality. Most probably recessive responsible for lethality. For example, very first that blue body color of mice is a lethal one. So early they did so many experiments. The first experiment is done with the help of mice, with the help of a mice. So at that time that mice is after delivery, after birth, immediately that mice will death it. Small mice will be death it. That is the example for lethal gene. Gene of yellow body color of mice is a lethal. That question may ask, ma. Lethal gene example, body color in mouse. Yellow body color of mice. Yellow body color of mice. That is the example for lethal gene. That is an important question. Yellow body color of mice. That is the example for lethal genes. The next one. Intergenic interactions. Next one, intergenic interactions. So inter means just now I told interaction between the different chromosome, different one. That is called as a intergenic. That is called as a intergenic. Here, interaction between genes on different chromosomes. Interaction between the genes of different chromosomes. In intragenic interactions, the genes of same allele. Interaction is occurs in the genes of same allele. But in intragenic interactions occurs between the different chromosomes. If one chromosome is here means there one gene will be present it. Similarly, another one chromosome is present means there also one particular gene will be present it. So that reaction will be occurs between the two like that. So interactions occurs the genes of different chromosomes. So what will happen after interaction? Phenotypic changes. Phenotypic changes. So interactions between the two genes on different chromosomes producing changed phenotype. Producing changed phenotype. Either intragenic interaction or maybe intergenic reduction. The result is a same. Result is a same. Phenotypic changes. Clear? Phenotypic changes because the gene is not interacted. Gene is not interacted means there is no changes. If one gene is interacted with another means that nature will be changed. So automatically phenotype will be changed. So here also end up that result of intragenic interactions changed in the phenotype. That intergenic interactions includes the 4 types. What are the types comes under the intergenic interactions means epistatic genes. First one is epistatic genes. Second one is duplicate genes, which is also known as pseudo alleles. Third one is complementary genes. Fourth one is supplementary genes. That 4 genes comes under the intergenic interactions. We'll discuss one by one. First one is epistatic. Epistatic means when genes prevent the expression of another non-allelic genes. The one gene is prevent the expression of another non-allelic genes means which is called as epistatic. Again, I repeat, when a gene prevents the expression of another non-allelic genes, which is known as epistatic gene. That phenomena is that prevents the expression of another non-allelic gene. That whole phenomena is called as epistaticity. Epistaticity. For example, hair color in dog. Epistaticity. Example is hair color in dog. Second one is duplicate gene. Duplicate gene means 2 pairs of non-allelic genes. 2 pairs of non-allelic genes is required for a characters. If any one of the gene is dominant, then this character is expressed. Again, I repeat, 2 pairs of non-allelic genes are required for determining the particular character. If any one is dominant, that character will be expressed. If 2 pairs are very important. Among the 2, any one is dominant. Means another should be a recessive one. So any n is dominant, it can able to express it. Clear? So that genes are called as duplicate genes. Example, fruit shape of Shepard's pose. That is a plant herb. Fruit shape of Shepard's pose. That is a herb variety. Next one is a complementary gene. So complementary genes means more or less similar to duplicate genes, but it having a major difference from the duplicate genes. However, 2 pairs of non-allelic genes are essential in dominant form to produce the particular characters. 2 pairs of non-allelic genes are essential in dominant form to produce the particular characters. But duplicate genes, what we discussed, 2 forms of non-allelic genes are required. Either one is dominant, that character will be expressed in the case of duplicate gene. But in the case of complementary genes, 2 pairs of non-allelic will be essential. The 2 pairs should be a dominant one. Then only that character will be expressed. That is called as a complementary. In duplicate, any one is dominant, it will be expressed. But in the case of complementary genes, 2 pairs of non-allelic genes are essential in dominant for character expression. That is called as a complementary genes. For example, flower color in Lathyrus odoratus. For example, in Lathyrus odoratus means a sweet pea plant. So flower color in Lathyrus odoratus, sweet pea plant. The last one is a supplementary genes. The last one is a supplementary genes. Supplementary genes, also known as a epistatosis. Supplementary genes, also known as a recessive epistatosis. Also known as a recessive epistatosis. Here, a pair of gene changes the effect of another non-allelic genes. A pair of gene can able to change the another non-allelic genes. Such genes are called supplementary genes. The one particular pair of genes can affect the another non-allelic genes. So one pair of genes can affect the another non-allelic genes, which is called as a supplementary genes. For example, coat color in mice. For example, coat color in mice. That's all about the gene interaction. Next one is a sex determination. Next one is a sex determination. So sex determination means, so the particular chromosomes are responsible to determine the character, whether it is a male or maybe a female. The particular chromosomes, particular sex chromosomes are most important to determine the sex, whether maybe a male or maybe a female, which is called as a sex determination. In sex determination, sex chromosomes are important. So sex chromosomes only determine the sex in human beings or maybe any other things. So sex determination means establishment of sex through differential development in an individual at early stage of life is called a sex determination. Establishment of sex through differential development in an individual at early stage of life is called sex determination. What are the mechanisms in sex determination means so many mechanisms will be involved, that sex determination mechanism will be vary from the each and every organisms, each and every living organisms. So many organisms, some organisms follows a XY type, some organisms follows the ZY type. Like that, that mechanism will be vary from the every organisms. Clear? The first one is a XY type. XY type. Here, male is a heterogamy. Male is a heterogamy. Hetero means we know very well, different one. Gamete means we know very well, which is responsible to form a zygote. So male is a heterogamy. What are they? X is a one type, Y is a another one type. That's why which is known as a heterogamy. Female is a homogamy. Homo means a similar. That same gamete will be repeated. That is called as a homogamy. XX is a homogamy, which is present in the female one. Here, that male only determined the sex. Here, male only determined the sex because the male only heterogamy. So, which gamete is present in which place, that person only can able to determine the sex. Clear? Here, heterogamete is the male. So, male only determine the sex. Clear? So, gynosperm. Gynosperm means a X chromosome containing gametes are called as a gynosperm. Androsperm means a Y chromosome containing gamete. Androsperm means a Y chromosome containing gamete. For example, human. So the two things are important. What is the meaning of gynosperm? What is the meaning of androsperm? Gynosperm means a X chromosome containing gamete. Androsperm means that gamete containing the Y chromosome. That is called as a androsome. Andro means present in male in high quantity. Example, What are the example of following the XX, XY type mechanism means human being. Human being, everybody's know. Totally, we have a 46. Among that 46, the 2 is important one. That one is the sex chromosome. One is a male having the XY, female having the XX. So human being, we are following the XX, XY type. Drosophila mammals. That example is important. Ma. They may ask in Neet exam. Which organisms follows the XX, XY type? Human, Drosophila, and mammals. Next one is a ZW types. That is a direct opposite to the XX, XY type. However, the male is a homogenic. In XX, XY type, male is a heterogenic, but in ZY type, male is a homogenic, means ZZ. Female is a ZW. Female is a ZW, heterogenic. So here sex is determined by the female one because heterogenic, heterogamete is present in the female only. So that's why that sex is determined by the female one. Birds, fishes, reptiles, butterflies follows the ZY types. Clear? So the here also that examples are important one. ZW types of mechanism follows the birds, fishes, reptiles, and butterflies. Next one is a XX, XO type of sex determination. Next one is a XX, XO type. XX, XO type of sex determination. Here, deficiency of one chromosome in male. Deficiency of one chromosome in male. That is called as a XX, XO. O means we can consider 0, like a 0. XX, XO type of sex determination. So deficiency of one chromosome in where? In male. The deficiency occurs in the male gamete only. So here male is a heterogamete. Heterogamete means at another gamete is absent, means O. So that's why which is also considered as a heterogametic. Clear? So male is a heterogametic. Female is a XX, homogametic. This XX, XO type of sex determination mechanism follows grasshopper, cockroach, and ascaris. And ascaris. Grasshopper, cockroach, ascaris follows the XX, XO type of sex determination mechanism. The next one is a haplodiploid. Haplodiploid. Haplo means a N, single N. Diploid means a diploid. Di means a 2, opposite to haploid. Clear? So haploid means a single N, diploid means a 2N. So haploid, so single N is present in one thing, and double N is present in the another one, which is called as a haplodiploid. So one sex is haploid. One sex is haploid. For example, male. So male is a haploid one. Female is a diploid one. So one is a haploid means another opposite sex should be a diploid one. So female is a diploid one. So this is called as a haplodiploid. So for example, honeybees follows the haplodiploid mechanism. So female, that's why in honeybees ants, mostly that female progeny will be produced more numbers because of the female only having the 2N. So female is the 2N in nature. That's why the mostly the in honeybees, the females will be produced more numbers. That is an important question mark. So honeybees follows a haplodiploid. That is important question. We may expect in Neet. That haplodiploid also occurs in ant. Listen, ma. So first one is a XY, XO, XX, XY, which is occurs in humans Next one is that is male is hetero, female is homogenic. Next one is ZY type. That is reverse order. I mean, male is homogenic, female is heterogenic. Next one is XXXO. One gamete is a lack in male. One gamete is lack in a male. The female is a homogenic. Clear. So that is called as a XXXO. In haplodiploidy, male is a haploid. Another one is a diploid. Clear. So next one is a mutation. Next one is a mutation. So what's the meaning of mutations? So everybody's know mutation means a sudden changes. That is called as a mutation. Mutation is a sudden changes occurs in the gene or chromosomes from one form to another form. One form to another form. The mutations occurs in the sudden changes of genes or chromosomes from one form to another form. That is called as a mutation. Sudden changes. Why the suddenly changes size occurs? Due to the any other radiations or maybe any other action of chemicals. So mutation is also defined as sudden heritable changes in genetic material of an organism is called mutation. So the mutation is occurs in the gene. Mutation is occurs in the gene. In gene level mutation is occurs, automatically it will be transferred. Automatically it will be transferred. Normally the genes will be transferred to onto another generation. So at that time that mutation also will be transferred. So you know very well, normally the character will be transmitted from one to another generation through the genes only. In case mutation is occurs in the gene means that while the character is transmitted from one to next one, at that time that mutated gene also will be transferred along with the character from one to next generation. Clear? So that mutated gene also, that mutation also heritable. Clear? That mutation also heritable. So sudden changes or sudden heritable changes in genetic material. Genetic material means the genes, DNA of an organism, which is called a mutation. So the term mutation was very first introduced by De Vries. Very first introduced by the De Vries. That is important one. I wrote in green color, De Vries. So you should know the important terminology of mutation. So mutation means you know very well sudden changes. Next one, mutant. Mutant means a unit of mutation. Mutant means a unit of mutation. That is called as a mutant. Mutant means a unit of mutation. So mutant is the smallest part of DNA which undergoes mutation. So smallest part of DNA which undergoes mutation. That is called a mutant. Next one is a mutagenesis. Next one is a mutagenesis. What is the meaning of mutagenesis? Mutagenesis means a process of mutation. That is called as a mutagenesis. Mutagenesis, Genesis. Mutagenesis means a process of mutation is called as a mutagenesis. Next one is a mutant, M-U-T-A-N-T. M-U-T-A-N-T, mutant. Mutant means which organisms carries the mutation. That organisms are called as a mutant. So mutant means which organisms carries the mutation. That organisms are called as a mutant. The next one is a mutagens. So mutagens are some substances can cause the mutation, which is called as a mutagens. So mutagens simply called as a mutation causing agent. Mutagens are mutation causing agent. That is a very important terminology. What is the meaning of mutation, mutant, mutagens, mutant, mutagenesis? That 5 things are important because anyone may expert in Neet questions. Again, I repeat, mutation means sudden changes. Mutant, T-O-N, M-U-T-O-N. Mutant means a unit of mutation. Mutagens means a mutation causing agent. Mutant means a organism carrying mutation. Mutagenesis means the process of mutation. That is called as a genesis. Genesis means a synthesis. Synthesis of mutation. That is called as a mutagenesis. Clear. So 5 important terminologies important in a mutation. So next one is what are the types of mutation? What are the types of mutation? The mutations are broadly classified into the mutagens, because before entering that mutation, we should know the mutagens. What are the types of mutagens? Because the mutagens only cause the mutation. So we should know the mutagens. Then we can enter the mutation process. Clear. So what are the types of mutagens? Means 2 types of mutagens are available. One is a physical mutagen. Another one is a chemical mutagen. One is a physical mutagen. Another one is a chemical mutagen. Chemical mutagen. So physical mutagen. So physical mutagen means physically, visibly. That is called as a physical mutation. For example, temperature. How the temperature comes under the physical mutagen? Because that is temperature, light, sunlight consists of the ultraviolet rays. Sunlight consists of the ultraviolet rays. That's why the temperature also one of the important factor, which is also comes under the physical mutagens. Clear. Then next one is a low and high energy radiations. Low and high energy radiations like X-ray, gamma rays, alpha, beta rays, ultraviolet rays, infrared rays, and so many rays. All rays are comes under the physical mutagens. Physical mutagens. For example, ultraviolet rays, infrared rays are comes under the low energy radiations. Remaining alpha, beta, gamma rays, X rays are comes under the high energy radiations. Clear. So now we'll discuss how that physical mutagens can cause the mutation. So that is called as a mode of action. Mode of action means how that physical mutagen can cause the mutation in the particular organisms. For example, first one is in mode of action also will be classified into the 2 types. One is the ionizing radiation, another one is the non-ionizing radiation. Ionizing radiations and non-ionizing radiation. First one is the ionizing radiation. So ionizing radiation means powerful mutagenic agents. Powerful mutagenic radiations are called as ionizing radiations. That ionizing radiations are high energy radiations. Just now I told ionizing radiations are high energy radiations. Here, that due to the high energy, that penetrating capacity also high. Penetration means it can able to enter that entering capacity, that penetration capacity also high, because which is a high energy radiations. Hence, due to the high energy penetrating capacity, it can able to attack the DNA itself. It can able to attack the DNA itself. Listen, that green color, that is called as a cell wall. So each and every organism having the, except animal cell, each and every organism having the cell wall. So inside that plasma membrane, after that cytoplasm will be present, then many organelles are floating in the cytoplasm. That nucleus will be present in the center part of cell. Nucleus will be present in the center part of the cell. You consider this is a nucleus. Inside that nucleus, DNA will be present. Inside the nucleus, DNA will be present. So here, ionizing radiations can attack DNA directly, because just now I told which having the high penetrating activity. So when we are applying that high radiations, so at that time what will happen? That radiation, it is a high energy radiation. So at that time easily penetrate the cell wall, plasma membrane and nuclear easily penetrate the cell wall, plasma membrane and nuclear membrane. In case of animal cells means easily penetrate through the plasma membrane and the nuclear membrane and attack the DNA, because DNA will be, or gene will be present in the nucleus. So it is a high energy radiation. So penetrating capacity also high. So hence it can able to penetrate the nucleus and attack the DNA directly and attack the DNA directly. That's why which is called as a direct hit. Direct hit. Direct hit. That mode of action is a direct one, which is called as a ionizing radiation. Ionizing radiation. What are the races are comes under the ionizing radiation means so many things. For example, just now I told alpha, beta and gamma races, alpha, beta, gamma and X-ray. Alpha, beta, gamma and X-ray races are high ionizing radiations that may ask in exam. So whatever may be in physical mutagen or maybe a chemical mutagen, it will cause a mutation. How means it will form a mismatch base pair. Mismatch means A should be paired with the T. I mean, adenine will be paired with the thymine, guanine will be paired with the cytosine. But instead of that thymine pairing, that adenine may be paired with the guanine, or cytosine may be paired with the thymine mistakenly. These things happens due to the mutation. That is called as a mismatch of base pair. Otherwise, thymine dimer may be formed. Thymine dimer will be formed. Mostly thymine dimer or uracil dimer will be formed. In case of DNA, thymine dimer. Dimer means a 2. Thymine means one of the nitrogenous base. Thymine. Clear. So thymine dimer means so one thymine will be bind to the one adenosine. But instead of that single thymine, two thymine may be bind with the single adenosine A. Clear. Single adenine. So one thymine will be bind to the single adenine, but that is a rule. But instead of that one, two thymine will be bind to the single adenine. So that is called as a thymine dimer. Thymine dimer. Why the thymine dimer is formed means thymine and uracil. So why sir that guanosine dimer or maybe a cytosine dimer is not formed means uracil and thymine is sensitive to the races. Only the two bases are sensitive. One is a thymine and uracil is sensitive to the races. So during that replication time, it will be thymine dimer will be formed. It clear. Next one, UV races also causes the deamination. Deamination means amino group will be removed. Amino group will be removed. That is called as a deamination process. Deamination process. Next one is a non-ionizing. Next one is a non-ionizing. Non-ionizing radiation. So non-ionizing means the science is very easy. Already I told you, we should see the literal meaning. Mostly 80% one is here means another one should be a opposite one. For example, night means that is opposite for night, day, like that. So in science, if you can, monohybrid means a dihybrid. One is a dominant means another one is a recessive, like that. One is a non-ionizing radiation means another one should be a non-ionizing radiation. Non-ionizing means a low energy radiation, because ionizing is a high energy. So it is a low energy. Ionizing radiations are a very strong, high mutagenic agent. Just now I told. But non-ionizing radiations are a weak mutagenic agents. It's like that you can compare. While you are studying, you have to compare it. At that time, easy to remember it. Clear. So weak mutagenic agent. So non-ionizing radiations are a low energy radiations. That's why which is a weak mutagenic agent. Weak mutagenic agent. What are the examples for non-ionizing radiation means ultraviolet rays and infrared rays. Infrared rays. Here, you I think now you are understood. Here penetrating capacity also low compared to the high energy radiations. Here, clear, because the penetration capacity is low. So that's why it cannot penetrate the nuclear membrane, Hence, it cannot attack the DNA directly. So when that changes occurs in DNA, at that time only which is considered as a mutation. Clear? When the attack the DNA or gene, so at that time only which is considered as a mutation. Clear? But here, due to the low penetration capacity, it will attack only the cytoplasm. It will attack only the cytoplasm. That cytoplasm brings the DNA changes. So here also the changes, sudden changes is occurs in DNA level, but the changes occurs indirectly. The changes occurs indirectly because here it while the ultraviolet rays attacks only the cytoplasm, which brings the changes in the DNA. So which is called as an indirect one. Here the DNA is not directly attacked. That's why it is called as an indirect hit. That's why it is called as an indirect hit. So this that's all about the physical mutagens. The next one is a chemical mutagens. Next one is a chemical mutagens. So chemical mutagens means it is more harmful than the radiations. So chemical mutagens are more harmful than the radiations. Means physical mutagens. So chemical is more harmful. The source of chemical mutations, where we are getting that chemical mutagens, where means through food. While we are taking a food, at the time rice, vegetables, we are taking a rice or vegetables. So at that time, the farmers may use pesticides, several pesticides, herbicides, or etc. etc. So at that time they are sprayed as chemicals. So while we are at the time they are sprayed the chemicals. So the chemicals will be present in the food, rice or fruits or vegetables. While we are eating, at that time that chemicals also entered into our body. So it can able to continue if you are continuously, if you are taking food means continuously taking that one means what will happen? What will happen? It may cause a mutation like air. Some gases, chemicals are mixed with the air. While we are inhaling, while we are respirating such air, at that time also it can able to cause a mutation. The next one is the water. Similarly, that chemicals are, for example, in a laboratory side. So while you are going to do MBBS or other higher studies, BDS, so at that time in practical section, you should use so many chemicals. So after completion of that practical section, you should discard that chemical in zinc. So automatically it will dissolve. It will be deposit in the groundwater. So if you are taking in that water means continuously, for example, dyeing industry, and so many sugarcane industry or textile or maybe so many effluent industries are they are releasing the so many effluents will be dissolved or will be mixed with the river water and groundwater while we are taking that, which consists of the many chemicals only. Clear? So when we are taking that water, automatically it will be ended. That's why I'm saying the sources of chemical mutagens are food, air, water, and other sources. Clear? The first one is what are the example of chemicals means carbon tetrasulfide, nitrous oxide, shortly known as HNO3, ethyl methyl sulfonate, methyl methane sulfonate, formaldehyde, several pesticides, phenol, TTT, diphenol, tetrachloroethane. The DTT is important one ma. Similarly, mustard gas. I wrote in green color. That is also important. Why means that is a first identified chemical mutagens. The mustard gas is a chemical mutagens. Very first mustard gas as a identified as a chemical mutagens. So that is also expected Neet question. I will explain the mode of action. Then I will close the session, ma. Mode of action of chemical mutagens. Mode of action means mostly it will be act as some that chemicals are act as a alkylating agents. Otherwise, that chemicals act as a base analogs. Otherwise, alkylidins. Otherwise, deaminizing agents. We will discuss one by one. The first one is alkylating agents. Alkylating agents means donating the alkyl group. For example, methyl group or ethyl group will be donating to the bases of DNA. Because that mutation, what's the meaning of mutation? Mutation is occurs in the genes or chromosome. Means first one is a chromosome. Chromosomes are present in the nucleus. Clear. The first one is a nucleus, chromosome, then gene, then DNA. The DNA is present in the gene. Genes are present in the chromosome like that. So last one is a DNA. So DNA consists of the base pairs. So that methyl ethyl groups, where the alkyl group will be donate means when that chemicals are present in the particular organism. So at that time, it donates a methyl group or ethyl group. That is called as a alkyl group. It will donate the alkyl group, either methyl group or maybe ethyl group. At that time, what will happen means it alter the base pairs. For example, alter the base pairs. AT means AG, that AC, that base pairs will be a alter. In case the base pair is altered, means automatically that phenotypic character also altered. Otherwise, we may get some disease. We may get some disease. That clear. This is called as a alkylating agent. For example, mustard gas, methyl sulfonate, ethyl methyl sulfonate. Mustard gas, methyl sulfonate, ethyl methyl sulfonate acted as a alkylating agent. The second one is a base analog. Second one is a base analog, which are similar to the DNA bases. Here, base analogs means some chemical structures. Some chemical structures are similar to the any one of the nitrogenous bases. Structure is similar to the any one of the nitrogenous bases. Here, due to the structural resemblance, for example, 5-bromyl rosyl, which is shortly called as a 5-BU, cas B small u, 5-BU, which is similar to the thymine, which shows a structural similarity. 5-BU is similar to the thymine, the structure is similar to the thymine structure. So that 5-BU will be act as a mutagenic agent. 5-BU means a 5-bromyl rosyl, shortly called as a 5-BU. This is structurally similar to the thymine bases. So I explain here the normal gene. So in normal gene, the first one is a will be paired with T, G is paired with C, C is paired with G, G is paired with C. The triple bond will be present in the GC, double bond will be present in the A and T. Due to the replication, so you know very well the when that mistakes are when that mistakes will be happens, how it will be transferred through the replication only. So here base analog, you can see here here that due to the structural similarity, the thymine is replaced by the chemical 5-BU. 5-BU. So the structurally it changed the DNA. Structurally it changed the DNA. That is called as changes is occur. That is called as a mutation. That is called as a mutation. So this is called as a base analog. Base means the particular base is altered by the action of chemicals. That is called as a base analogs. Next one is third one is acrylamide dyes. Acrylins. Acrylides means dyes. Just now I told so many dyeing effluents or so many things will be releasing the effluents in water. So at that time, which consists of the many dyes, or maybe we are using the many dyes in laboratory size. That's dyes such as so many dyes we can use it. For example, proflavin and agridine orange. Those are the some important dyes. Proflavin and agridine dye. Those are the important dyes which cause disorders. Which cause a deletion or insertion. So acridine, which are dyes such as proflavin and acridine, which is a dyes which can cause the deletion or a insertion. For example, this is a normal genes. This is a normal genes. Through mutation, while the DNA is goes a replication process. At that time, the last you can see the last one, AT, GC, CG. The last TA here you can see Here you can see that TA is present here. T is present in another one stand. A is present in another stand. That TA. Here you can see the TA is deleted. Clear. So that what is the action of accordion means that one base means any one a adenine or thymine, that is called as a base pair means adenine paired with thymine or guanine paired with the cytosine, that is called as a pair means two. That base pair will be deleted or base pair will be inserted by the action of accordion. At that time that gene structure DNA structures will be very automatically, it will express in the phenotypic traits. That is the action of accordions. The next one is a deamination agent. Next one is a deamination. Deamination means a D means a removal. Amino means that removal of amino group. That removal of amino groups. That is called deamination. So mostly nitrous oxide, that is an important question. Nitrous oxide causes a mutation. Nitrous oxide causes a HNO3. Nitrous oxide causes a mutation by deamination of DNA bases. So mostly the deaminations will be caused in adenine, guanine, and cytosine. Adenine, guanine, and cytosine. It won't cause a thymine. Clear? So here adenine group. So you know very well in DNA structure, in bases structure, adenine, guanine, thymine, cytosine. Bases structure, if you are seeing means one is amino group and another one is a hydroxyl group will be present. You know very well that amino group will be replaced by the hydroxyl group. See, this is a structure of adenine. This is a structure of adenine. Here that amine group will be present. Amino group will be present. That amino group will be replaced by the hydroxyl group. That amino group will be replaced by the hydroxyl group. Amino group means NH2 will be replaced by the hydroxyl OH group. Clear? So at that time, that nature will be changed. That adenine becomes a hypoxanthine. Adenine becomes a hypoxanthine due to the replacement of amino group in the 6th position. So the 6th position of amino group of adenine will be replaced by the hydroxyl group, OH group. Clear? That deamination of adenine converts AT pair into the GC pair. So what will happen, sir? In deamination means deamination, due to the deamination, that adenine converts the AT pairs become the GC pairs. That convert AT pair into the GC pair. Clear? So those are the important action of mutation. So time is not enough to complete mutation. So now we discuss up to mutagens. So today we discussed about the linkages, sense linkages, and in mutations. In mutations, we discussed the terminology, important terminology of mutation and mutagens. Clear? So regularly I am telling, so you must recall it. So recalling helps to score a high score. Clear? So we will meet in next class, ma. Tomorrow we will meet, ma. Thank you. Today class is over. Thank you.